talking, you should just do this.
Oh, I couldn't find the mouse. Where is the mouse? Can you see it?
It's on the other side.
Okay. And then how? Okay. Hypothetically, I am just clicking. Oh, okay.
It's just on the second screen.
PDF, please. Yes. Control that. Oh, okay, great.
So there's a slide advancer if you want,
Okay, then I don't.
or there's also
Okay, thank you.
laser one.
Okay.
This is off if you want. Just
Good afternoon, everybody. I'm Beste Turanli. I'm a PhD student at KTH in Sweden. Before I start, I would like you to thank organizers for having me. And my presentation name is Model-Driven Discovery of Drug Targets for Effective Treatment of Prostate Cancer. So today I will start with a short intro. Oh, sorry that I didn't check the screen. Okay. Sorry again. So today I will start with a short introductory part about the concepts and the terms that I use in the study. And then my project has a two main parts. First part is about reconstruction of the model and the phenotype evaluation, and second part is about drug repositioning. And then I will talk about the conclusions that we get, and I will finalize the presentation with the future studies that we are planning to do. So this study is a system biology study. Actually, what is systems biology? As you all know, the complex system has many integrating subsystems with many integrating components in a particular environment. To study and understand this complexity is always a challenging task. In my case, the complex system was prostate cancer, and I used genome-scale metabolic models as a tool to understand this problem and repurpose a drug. So the current state of the prostate cancer, we all know that cancer is a global health problem, as you can see on the left on the World Cancer Incidence Map. And prostate cancer is the recent cancer statistic by American Cancer Society reported that prostate is the first leading size of the estimated new cases and second for the estimated deaths in 2018. So genome-scale metabolic models, in my humble explanation, I can say that genome-scale metabolic models are biologically meaningful mathematical representation of the metabolism. So if you would like to construct a model, first thing that you need to have data about the metabolism as much as you can. You can go and check the biochemical reactions through the literature, or you can use previously constructed models, publicly available omics data or patient-specific data, as well as the environmental data. After you get all this data and the processing part, we also need to construct a cytochromatic matrix to represent these biochemical reactions. And integration of the phenotypic information, we are getting a genome-scale metabolic model to be used for the clinical validations, identification of biomarkers, for diagnosis, or for drug discovery. But drug discovery is a long way to go. So nowadays, we are also focusing to find some drug targets using drug repositioning techniques. So it's also called drug repurposing or drug recycling. Basically, it's identification of new therapeutic applications of a known drug. So it's, since no drugs has established formulations or manufacturing methods, known adverse effects, so this method is cheaper, faster, and less risky comparing to conventional drug discovery methods. In this study, first thing that we aimed, reconstruction of a generic genome-scale metabolic model for the prostate cancer. And second thing, repurpose a drug for the prostate cancer treatment. And the third thing that we would like to do, test these drugs' effects on tumor growth via the genome-scale metabolic model that we reconstruct. So first thing that we did, reconstruction of the metabolic model. For this purpose, I used personalized genome-scale metabolic models. It's 470 different models. I unified all these personalized models at once. Then I also integrate prostate cancer tissue-specific proteome data using the TNIT algorithm. And in the end, in my model, there are 2,655 genes and 6,718 reactions. And I use this genome-scale metabolic model in the essentiality analysis. It's basically in silico gene knockout. I checked, first thing that I did, logical transformation of the model is kind of pre-processing step. And then I set the objective function as a biomass maximization since we are dealing with a tumor. And after the simulation, I get 22 synthetic lethal genes. So basically, when we knocked out these genes, this prostate cancer model is not growing, tissue is not growing. All these genes are, most of them are enriched steroid biosynthesis, and unsurprisingly metabolic pathways, biosynthesis of antibiotics, terpenate backbone biosynthesis, glycerophospholipid metabolism, and peroxisome. On the other hand, the second analysis that I did was a drug analysis. It based on the normalized gene expression from non-tumor and tumor samples. And what I would like to see, which pathways are tightly connected on tumor samples, not on a non-tumor samples. So it's also ended with the steroid biosynthesis again. It's highlighted. Ethyl lipid metabolism, glycosphingolipid biosynthesis, riboflavin, pentose phosphate, and thyroid cancer was highlighted after this study. So I was expecting to see something about maybe steroid biosynthesis or lipid metabolism related to lipid metabolism. And the second part of the study was drug repositioning part. Now we got a reconstructed model, and I have 200 and 600 genes, as I mentioned. And on the other end, I run differential expression analysis, and 370 differential expressed genes that I have. When I check them, intersection of these two was 641 differential expressed genes from the model, and we call them metabolic gene signatures. And we use them as a query in connectivity map 2. It's the conventional algorithm that's already used in connectivity map studies. So first thing that we did, we calculate similarity scores based on the modified Kolmogorov-Smirnov test, and then statistically significant drugs based on the random permutation. And I only take the drugs with a similarity score below than 0, because I was trying to find which drug has a reversal effect to my metabolic gene signatures. So it's the first half of the apple that I would like you to keep in your minds. So if I would like to test these drugs in my model, I also need to know which genes has interaction with these drugs, or which drug has interaction with this gene, with genes. That's why I also interpret the connectivity map data to repurpose novel gene-drug interactions. I got the raw expression data for 3 cell lines, HR60, MFC7, and PC3. And I choose a single treatment with the highest concentration, and which has also highest correlation within this 3 cell line. And for the control cases, outliers based on the hierarchical clustering were excluded, and the mean gene expression was computed across the controls. And in the end, 683 treatment instances and the single control per cell line that I had. Then for each cell line, log fold chains were computed by comparison with the respective control. And then I calculate the confidence score for these drug-gene associations. For these, first I calculate p-values, and I use all these p-values from different 3 cell line. I use Naive-Bayesian formula to calculate one consensus confidence score. And then metabolic gene signatures, I mapped these metabolic gene signatures on the drug gene metrics that we get with the log fold 2 chains and confidence score. And in the end, I found 225 upregulated genes and 301 downregulated genes across to 683 drugs. And it's the second half of the apple. So when I merge these 2 half pieces, it concluded with the 23 candidate drugs. And I checked the literature if these drugs are already associated with prostate cancer before, and 12 of them were already tested on prostate cancer or only hypothetical computational predictions, but they were all, when I did the text mining, I see that 12 of them are already associated, and 11 of them were not associated yet. And then I go back to my model that we reconstruct. I did in silico cell viability tests, and I used this all 23 drugs with the interacting genes with a high confidence score. and then 8 of these 12 associated drugs had an inhibitory effect through the model, and 5 of the 11 not associated drugs are also showed inhibitory effect after the analysis. So, as a summary, I started with the 1,309 drugs from connectivity map 2, and while I was trying to find drug-gene interactions, I found 683 drugs. And after these KA tests and random permutations, it decreased 23. And after literature review, it was 11. After the in silico cell viability test, there were 4 candidates. We excluded once because it was veterinary drug. So we go with these 4 only. As a conclusion, what we did, we reconstruct a genome-scale metabolic model for prostate cancer cases derived from individual transcriptome more than 400 and prostate cancer tissue-specific proteome data. And we explore which genes are essential and which pathways tightly connected in prostate cancer. And we did an interpretation of drug-induced transcriptome data to reveal new gene-drug interactions. In the end, we propose 4 different drugs: sulfamethopriadazine, azacilin, hydroflumethiazide, and ifamprodil for the treatment of the prostate cancer based on the in silico cell viability assay that we did. And we directly started with the experimental things. And the first drug that we try, ifamprodil, and shows a inhibitory effect on the in vitro cell line on PC3. And what can we do in the future? For sure, we can do in silico validations, like molecular docking for the drugs and their target genes that we assume. And we can use the same pipeline on independent prostate cancer data set. And we can do in vitro validation as we are doing now. But we can also test the different prostate cancer cell lines. And we can test the cell toxicity or revealing the drug mechanism. And for sure, in vivo validation is really important to see systemic effect, or therapy options with the drug delivery systems can be done using these drugs. So thank you for your attention. I also would like to thank Student Council or SysMod for the travel grant and the financial supporters of this project. Thank you. Thank you.
I was wondering if you could elaborate on how proteomics and transcriptomics data are combined before generating the contextualized gen.
You are asking me.
Can you repeat the question?
Yeah, sure. You are asking how I use transcriptome and proteome data at the same time as a signature while I was reconstructing the model, right? Actually, the algorithm that I use is for automatic reconstruction of the metabolic model, so it helps me to do this. The thing that they did, so transcriptome data is just on the map, while we have a generic human metabolism network. I use HMR2 for this. And first thing that the algorithm did, mapped the transcriptome data first, and it's just highlighting these genes and associated reactions. And then it's also the same, doing this for the proteome. It's then proteome data is just filling the gaps. If I have. Hey, you're welcome. And I have a poster, by the way. I didn't put in my presentation, so we can also talk in detail. 2, 4, 3. There was another question. Then.
Arthur. Hi, terrific talk. I'm concerned that you're modeling just metabolism, and there's a lot of other activity in the cell.
Yeah.
And I'm wondering to what extent the activity that you're not modeling might influence the drugs that you're recommending be repurposed.
So you are asking, there are a lot of different interactions that we may exclude while just we are working on metabolism, right? So yeah, it may affect the results for sure. So it's also I can maybe integrate co-expression networks, signaling networks on this, but it's just a kind of, maybe we can assume that it's a preliminary, but the first thing that I would like to see was the metabolism actually, because whatever I did about this direct analysis or other analysis that I did, basically differential expressed gene analysis, is always focusing the metabolism for the prostate cancer. It's the first thing that I found. So that's why I think that maybe it's a good starting point to start with metabolism. That for sure it will affect.
Have you looked at the effect of these 5 drugs on healthy cells?
Through the model in silico, you mean?
Right.
Yeah, actually, I started to do this. Yeah, yeah, for in a human metabolic atlas on the webpage, also everyone else can get all these healthy tissue models, and there is around 30 tissue models, different 30 tissue, and now I am checking 56 different biological tests to see if there is a in silico toxicity of these models. Yes, I'm doing this now. Yeah. Thank you.